Great. Hi, everybody. We're at our webinar 3 for Microbiomes for All webinar series. I'm Rachel Horak. I'm your tech operator and host for today from the American Society for Microbiology. If you have questions during the seminar, you can put them into the chat. There will be times where the speakers will be able to allow you to unmute. So at the time that they ask for questions, raise your hand and you'll be able to unmute them. If you ever have questions during the session and you want to ask anonymously, then feel free to send me a private message and I'll ask it anonymously on your behalf. So now transcript is enabled. These are auto captions, so keep that in mind. And I'll turn it over to our speakers at this time.
Good morning, everyone. And everyone is excited for hopefully the third session. Let me just get up here and share the slides. Everyone can hear me all right? Okay. And can everyone see the slides? All right, excellent. So we're continuing on with the data analysis, and we've been pretty ambitious. And again, today is another ambitious day. We may not get to all of this, but what I'm hoping to be able to cover is the QIIME2 output for bar plots, which is really cool. And also how to go from that to heat maps and Venn diagrams in Excel, because these are really excellent tools to get students started with actually kind of manipulating data in Excel. And then we'll revisit PCOA plots, because QIIME2 has kind of slightly more kind of cooler PCOA plots than the Data2 output. And then we're going to touch on Microbiome DB. I'm going to show you one thing. That's a whole nother kind of platform. So we're not going to have a time to go into that. That might be something that we do a add-on session at a later point if there are people that are interested in that, but we'll touch on that. And then we'll switch over to Avram, who's going to talk about student assessment, and then we'll wrap up. And I was thinking this morning as we were hearing that William Shatner is going into space, I was kind of thinking of the Apollo 13 reentry scene as kind of a metaphor for data analysis, because I know to me now, and probably to some of you out there, all of these, different platforms, different, terminology, different data sets, different files, flying by you, and you're just hoping your heat shields are intact and you can make it through the atmosphere and then kind of parachute softly down to a beautiful ocean of data to analyze. So we know that this is tough. We know it's a steep learning curve. And we also know that this kind of 3-hour webinar is not necessarily going to be conducive to us kind of looking over your shoulder to see what's going on your computer while you're having a specific error pop up. These, hurdles are going to be different from person to person, from platform to platform, and so forth. And it's, hard to do that via webinar, but we'll talk about this more at the end, that we'll, we can work with people one-on-one. We can set up smaller Zoom sessions if there are a handful of people that want to go into more detail or get more guidance on certain elements. And so we're not going to kind of just leave you hanging after this webinar. We'll be sure to follow through. So don't panic, don't be upset if you're not getting as far as you want. We'll be able to circle back and bring people to where they want to be. And so with that, I think we're going to switch to a poll. Rachel, I think.
Hopefully you're all seeing a poll right now. We just need to know how many of you managed to get through the first stage. So Marianne, I hope you can see our poll. We're doing a poll right now just to see if you're able to manage to get the data that you needed. We've 22 respondents already. 5 more to go. Actually, that's probably us. Is that us? How many of us is there? Theo, Davida. I think that's everybody.
Sharing results.
There you go. Carla just joined as well. Thanks, Rachel. On we go. Thanks, Theo.
Okay, so we got that. All right. Again, this is the microbiome analysis pipeline. And just to reposition everyone, we're starting over here on the lower right-hand corner with the FASTQ files. And again, some of you may have your own FASTQ files from your own data sets. There are data sets that are available from other sites. And we are providing the demo soil data sets for anyone to use during the workshop or on their own with your students and so forth. So everyone should have a source for FASTQ files. And what we talked about last time were some of the outputs that come from Data 2 and QIIME 2. Those are both analysis pipelines that are built into Nephele. And so today, we're going to focus more on some of the outputs from QIIME 2, which we introduced a little bit last week, but didn't have a chance to go into in a lot of detail. So hopefully we can, I'm going to try to do this live, so hopefully the sites aren't too slow. So we're going to jump right into exploring the data. And the first file that I'm going to take you to is the taxabarplot.qzv. And so here you can see on the screenshot here that if you got your FASTQ files into Nephele and you chose to run one of the analysis pipelines, QIIME 2 was one of those options. And if you ran QIIME 2, then you got an output folder that looks like the folder here with, again, this whole list of different files. Last week, we looked at like the table.qzv. We looked at the alpha rarefaction.qzv. And just to remind you that any of the files that have a QZV are, that V is for viewable. And so you can view those in QIIME 2 view. So I'm going to switch over now and try to open this taxa.barplot QZV file. If I can. Oh, wait. Give me a second here. Okay. Can people still see my browser? Hopefully, is that a yes on being able to see?
Confirmed. Yes, we see it.
Okay, great. So here I am at QIIME 2 view. And remember, you can drag and drop. And you can also click here. And I don't think you can see my pop-up windows, but I'm getting a pop-up window to my data folder. And I'm going to I'm going to go to my output file from my single end analysis for QIIME 2. And I'm going to look for my taxa.barplot.qzv. And I'm just going to select that and open. Okay. And so this is what the first screen will look like when you're looking at the bar plot, and it looks a little bland. Okay, so what I want to show you first is you'll see this bar width slider here on the left. So I'm going to slide that to make my bars a little wider so we can see more. And then the first thing that I'll draw your attention to is the taxonomic level. Right now we're at level 1. Okay, that's the kingdoms here, archaea and bacteria. And so that's why it looks, just as monolithic bars of bacteria. Down here, you can see maybe a little bit of a archaea, but mostly bacteria. So I'm gonna toggle over to level 2, which are gonna be my phyla. And here it starts to look a little more interesting. And so here on my right, just one sec. Here on my right are the list of the phyla in the sample. And what I'm going to do is I'm going to sort my samples. Again, this populates automatically from my metadata file, and I'm going to sort by source. Okay. And what you can see down here is now my different samples are listed by the source that they came from. So if this is flower bed soil or quad soil or leaf litter, they're all listed there along the X axis. Other things that are worth noting here when you're looking at the bar plot view is that you can highlight, well, first of all, if you hover over the bars in the upper left-hand corner below that slide bar, the bar width slider, it tells you exactly what you're hovering over and what that is and the percentage, okay? So in this case, I'm looking at East Quad flower bed, and it's Actinobacteriota, and it's 35.821%, okay? So you can slide over these things and it'll show up in that upper left-hand corner. The other thing that you can do is you can click on phyla to have them isolated. So if you only want to look at the proteobacteria, you can click at that and it's just isolating the proteobacteria. If you want to look at the proteobacteria and the acidobacteria, you can click on the acidobacteria too, and you can get that listed as well. So it's a nice way to kind of highlight things within the bar plot. And if I just re-click everything, then I get back to my normal view. Usually I start at the phylum level because there's a lot of information there, but you know, you can obviously go down to class, order, family, genus, and species. When you go down to here's, it'll take a minute to generate, down to genus, it gets pretty colorful. And so I usually start at phylum because that's a little bit more manageable, but you have that option to go wherever you want. So I'm going to switch back to level 2. And I'm just going to pause. I'm going to move on to the next thing in a second, but I just wanted to pause to see if there were any questions in the chat or anything anyone wanted me to address.
Could you just very quickly just go back to opening the bar plot one more time very quickly from your output folder, Theo?
Oh yeah, so I'm going to start again at QIIME 2 view.
Thank you.
And I'm going to click here. I can drag and drop, but in this kind of when I'm doing a webinar, it's easier for me to just click here. And again, I don't know if you see my pop-up windows, but it's popping up my data folder. And I'm just looking for the text of barplot.qzv, clicking on that, and then it's opening. And then I'm just widening the bars as you know, again, depends on how many samples you have. There's 18 here. I'm just widening the bars, checking the taxonomic level, switching it to phylum, and I'm going to sort on source. And those things, like I said, autofill from your metadata file that you already uploaded in the first steps when you uploaded your data to Nephele. All right. Were there any other questions, David, in the chat or anything I should address?
Yeah, there is actually. Aaron Olsen wants to know, can you speak about accuracy when identifying genera and species level?
So that's going to vary a little bit depending on the length of your reads. So again, we're only doing single end reads here, so we don't have the full length that you would have if you had paired end reads. With these data, I'm very comfortable at the levels of, you know, phylum, class, order, family. Genus is okay most of the time. Species is iffy. So I'm comfortable if I'm looking at these data with students to say if we see, you know, different genera there, then I think that that's I'm confident in that. But I don't try to make too much out of the species level, if that's what you're asking. If I had paired end data here, I would probably feel a little more comfortable going down to species level. Anything else?
That's it for now.
Okay. So the next thing that I want to do, so you can save these. If you look over to the upper left-hand corner, you can save the bar plots in a number of different formats. But what I actually wanted to show you is the CSV file, because this is, there are a lot of ways to kind of get to these data, but this is one of the easiest ways if you want to get an Excel file that you can then go on to do heat maps. So I'm going to go directly from this bar plot data using the CSV file to doing heat maps and Venn diagrams, which are, again, things that are nice to do with, to get the students started and really get your hands dirty doing some of the data analysis. So I'm just going to click on CSV, and you can see down here in the lower left-hand corner, it downloaded this level2.csv file. Okay, and I'm just going to click on that to open it. And now I think I need to do a new share so you can see my spreadsheet. Can people see my spreadsheet now? Okay,
Yeah,
so this is what the data look like when they come down in the CSV file. And you have your samples down in column A. These are your sample identifiers. And then because I did this at the level of phylum, all of the first row 1 is all the different phyla that showed up here. And when I click on a different column, so here's K, this is Actinobacteriota. And if I click on column N, this is Bacteriodota, and so on and so forth. So all those phyla are listed across, in column 1. The first thing I'm going to do is I'm going to go down here and I'm going to create a new sheet. So this is one sheet within Excel. And I'm going to create a new sheet. And I'm just going to label that heat map. Okay. And I'm going to go back to my original sheet and I'm going to select all the data that's here. And you can see over here, the last columns are my metadata. Okay, so they get brought along in that download. So I selected everything. I'm copying it. And now I'm going to go back to that heat map sheet that I made. And I'm going to go to, from my pull down menu, I'm not sure if you see the pull down menus or not, but I'm going to click on paste special. Okay, so I select paste special. There are a lot of options here, but I just want to draw your attention to transpose. And I'm going to click the transpose box, and this is basically going to turn my columns and rows. It's going to switch them, and I think that that's a little bit easier to view. Okay, so now when I paste, everyone, hopefully you can see now the samples are in row 1 across the top, and the phyla are in column A down the left-hand side of the screen. Okay. So then the next thing that I'm going to do is I just want to get the phyla names because there's a little more information here than I want. So I'm going to select all the phyla. So that's rows 2 through 58. And I'm not going to take my metadata, just I'm going to go 2 through 58. I'm going to copy that. I'm going to slide over to some empty cells, and I'm just going to do a normal paste there. And so there are all the phyla, but you see the download automatically comes with my domain information. So that all has archaea or bacteria. And if I did this at class, then I'd have all that information. I'd have domain, phylum, class. Okay, it's just the way that the CSV file downloads from QIIME 2. So what I'm going to do is I'm going to select data, and I'm going to pull down that menu to text to columns. And I'm going to click on text to columns, and I'm going to say, yes, my columns are delimited. And I want, you can see there's some delimiters I can choose. there's tab, semicolon, comma, and so forth. And I'm choosing other because I want to split up my data after the underscore. So I've just put in underscore for other, and you can see in the preview what it's going to look like. So I hit next, and there's some more options, but I'm just going to skip these options because I don't need to worry about those, and finish. Okay. And so now it took all of that information and broke it up. And now I have a column that's just the names of my phyla. And there was one that couldn't be classified as anything other than bacteria. So I'm just going to fill that in as unknown. So now I have all my phyla names. I'm going to select those, copy those over, and I'm going to paste them back into column 1 and title this phylum. Okay, and then I'm just gonna, I don't need these, this other information, so I'm just gonna clear all that. And I'm gonna go down to my metadata, and I'm gonna look at source. I'm gonna copy my source data, and I'm gonna put that up along the top because that's a little bit more informative. So now I have my phylum down the column A. I have my sources across row 1, and I have the numbers. This is a number of times that each one of those phyla were counted in a given sample. And of course, looking at data like this, it's really hard to see any patterns. And so that's why people like to use heat maps. So what I'm going to do towards making this heat map is I'm going to select all of my numeric data. So those are all the values that are there. Okay, so I've selected those all. And I'm going to use conditional formatting. So I'm going over here, and it might be in a different place depending on what version of Excel you're using. And I'm going to select conditional formatting. I'm going to collect color scales, and I'm choosing the blue, white. And I'm actually going to go back and look at my color scales and select the more rules. And I want a 3-color scale. And I want my lowest value to be red. I want my midpoint to be white. And I want my highest value to be blue. And those may not be the colors that you see because I'm colorblind, but those are what the colors look like to me. And then I hit OK. And so you can see I'm kind of moving in the direction of heat maps. I've got some high values that are kind of darker blue, and then all my 0 values are red. Okay. So we can make this a lot nicer. I'm going to select all the borders. Okay. And I actually want to see the numbers. I don't want to see the values. So I'm going to go to Format, and I'm going to pull down for my cells option. And I'm going to choose the last option, which is Custom. And I'm going to just put in a semicolon here. And what this does is it's just a setting that Excel recognizes as keep the values, but don't show them. Okay. So my values are there, but they're, you can see when I click on them, they show up in the status bar up here, but they don't show up on the graph itself. And so now I have the nice colors. And what I can do to make things a little nicer is to angle my text a little bit. I'll select these so I can see everything nicely. And then for these columns, I'm going to format columns, and I'm going to change the width. And it's set to 10. I'm going to change it to 3. And so now it's starting to look a little bit more like a heat map. Okay. And then what I can do is I can select all my data. Now I want to see patterns, right? And right now the phyla are just listed alphabetically. So if I select my data with the phyla, I can go to data and I can select on sort. And my list has column headers, so I'm gonna leave that as my list has headers over here on the right. And I'm going to sort by, and again, I'm just gonna pick peninsula soil. And I'm gonna do largest to smallest. Okay, and now my data is sorted, so the values that are the largest are showing up at the top. Again, and this is based on peninsula soil, or at least the first peninsula soil sample. And you can see it starts from the highest and goes down to the lowest. So there's some more order here, and you can start to see patterns. Okay, now the last thing I'm going to show you is like a bonus feature. You don't necessarily have to do it, but I like to do it. I'm going to copy this over. Okay, I have a copy now. I'm going to reselect my columns. I'm going to format the column width again down to 3. Okay, so there's my pretty plot. It's identical to what I have to the left. But now, instead of doing these values, I'm going to put in a formula here, and I'm going to look at the log2 value of these numbers. Okay, and so the formula on the left is showing me I'm doing the log, and it asks me which cell do I want to perform that on. So that's the first cell in my data set. And then it's asking me what I want as a base, and I'm putting log base 2. Okay, and I'm just pasting that in there. And now I'm going to copy that. And paste it. Okay, and so now it's looking a little bit different, but you see there's a problem, is that you can't do a log on 0 values. Okay, so I'm going to go up and I'm going to edit my thing, and I'm going to cheat. I'm just going to add 1 to all of my samples. Okay, and I don't know if that's statistically kosher, but remember that the heat map is to see patterns. We're not making any important value judgments. Okay, so I'm going to repaste that. And there you have, when you do it on the log base 2 scale, you're basically on the left, you have a lowest value of 0, and your highest value is like 22,000 or something like that over here. Okay, this is your highest value, 22,993. And so that's a lot of breadth to try to get into one thing. It's a little bit too much. So if you do it on log 2, it scales everything down, and you get more kind of resolution, and you can actually start to see the patterns a little bit better. Okay. So I think that that's a nice thing to be able to do with the students. If you can get them to make a heat map like this, then you can start to ask questions. You can look at, say, the sediment soil, and you can say, well, sediments tend to be anaerobic. So if I look in my sediments, okay, do I see phyla that are enriched, that are associated with anaerobic bacteria? Or you can look and you can ask students like, okay, leaf litter, what about my leaf litter samples? Leaf litter, we might expect to find saprophytic bacteria. So you can look at your leaf litter, and you can look at what phyla are enriched, and you can see if there are phyla that are that have members within that phyla that are well-known saprophytes. Okay, so you can start to explore the data there. All right. So I'm going to pause there again to see if there are questions. I know that I'm working on Mac Excel, so it might be a little bit, some of the options might be a little bit different for whatever Excel you're using. So I may not be able to answer any of those kind of Excel to Excel variants, but I'll just pause for a second if there's any general questions about the heat map. Okay, so that's heat maps. Now, another thing that I think is that you can do with students that is interesting is to make a Venn
Yeah,
diagram.
there is a question actually, just before you go on. When you compare, is it qualitative or is there a way to normalize reads between samples?
So this is, yeah, so I believe that these data going into the QIIME2 bar plot are rarefied, so there should be some normalization there already. But you can either do that within the Excel sheet if you want, or you can just check your parameters when you're downloading the CSV file from the QIIME2 view. But again, I've just taken the file from QIIME2 view because it just followed naturally, but you can get the CSV file directly from the data2 output or the QIIME2 output. There's more than one place where you can get this this data, and then you can either just check in your pipeline to see if it did rarefaction or normalization. If it didn't, then you can do that within Excel before you make the heat map. But I didn't do that here. I'm using this as a tool to show the students the patterns that you can see, the bacteria that are enriched. You can see that our soils are full of actinobacteria and proteobacteria and acidobacteria. That's fantastic because there's a paper that came out a year or two ago that looked at all of the world's soils, and hey, guess what? All the world's soils are high in actinobacteria, proteobacteria, acidobacteria. So it's fantastic. Like, hey, you know, your data are in agreement with everyone else's, which is kind of exciting, and gives you confidence that you're going in the right direction. You know, again, these data I'm not using to make statements like Proteobacter is 5 times more abundant in this leaf litter sample as opposed to this houseplant sample. That's the heat map isn't, you know, ideal for doing that kind of a quantitative one-to-one comparison. Are there other questions, Avram?
Yeah, there is one. Karen Thickman asks, what would it look like if you arranged the columns based on the PCA results, on the principal components analysis results?
Right, okay, so I'm gonna direct you to the StatsQuest video on heat maps. What I haven't talked about, and what's a crucial factor of heat maps, is how you sort your samples and how you sort whatever parameter is that you're looking at. So I sorted based on phylum abundance, and that gave me the pattern where I could see the most abundant phyla at the top. But you're very right that many of the heat maps are going to use other metadata feature to cluster the samples. And I haven't done that, but you can do that. I've got them just clustered by you know, what type they are, but you can use phylogenetic measures or other metrics to cluster those things. So maybe you want all the soils next to each other, you know, and that will make it easier to see the patterns, because the really crucial point, which I think a lot of people miss, and this is an excellent question, is that if you don't have your axes sorted properly, it's harder to see the patterns. And so like when I don't sort for abundance, if you remember that first graph, the kind of the abundant sample, or the abundant squares and the zero squares were kind of all spread over. So the sorting is critical, but I would direct you to that StatsQuest video. I'm, you know, it's going to do a better job than I can, and I just didn't prepare to, you know, do that during this session because there were some other things I wanted to show, but it's an excellent question. Avram, is there anything else?
No, that's good.
Okay. Okay, so I'm going to show you Venn diagrams. So I created another sheet, and I just called it Venn, okay? And I'm going to go back to my original data. This is the CSV file that I downloaded from Chime 2. And I'm going to do what I did before, is paste special. And I'm going to do transpose. Okay. So here are all my data. I'm going to do that quick trick of text to columns by going to data, text to columns, delimiter underscore, and there are all my phyla. I'm going to paste those back here. Just re-header that. I'm going to go down here and I'm going to, again, do like I did before. I'm going to take my sources and I'm going to paste those across the top. And so I've got everything ordered the way that I want. And I need to now copy these data over. So I'm going to select and copy. I'm going to go over to some, I didn't delete this stuff, but I could have deleted it. I'm going to go over to a clear area. I'm going to paste. And so now I've got a copy of what I just, oops, copy of what I just had. I'm going to try to widen this column a bit so you can see the file. Okay, and now I want it for the Venn diagram. It's a binary thing. I don't need to know how many hits that I got in a specific phylum. I just need to know, was that phylum present or not? There's a lot of ways to do this. I'm just going to do it in kind of a quick and dirty way that seems intuitive, at least to me. And I'm going to put in a formula here. And I'm going to use an if statement. And I'm going to say if my value over here does not equal 0, okay, so that's my logical test, then what I want it to put in, I want it to just give me the name of the phylum back, okay? And if it is 0, then I just want it to leave it empty. And so if I just put those open, close quotes right next to each other, that'll give me empty, okay? And I can enter, okay? And so now that first value was 0, and so it's giving me back an empty screen. Now I just have to do one thing. If you haven't worked with Excel more, I need to lock this column. And if I need to lock a column or a row, I need to put a $sign in front of it. So now that column is locked. And I can just paste that formula across my whole thing. And now you can see, hopefully it takes a little while to populate. But now I've changed those values just to tell me that that phylum was there. And so now what I can do is I'm going to copy this first column, which is my leaf litter column, copy that, and I'm going to have to jump around screens a little bit. So I'm going to go back to my and I'm going to go to a program called Venn 2.1. Okay, and if you just Google Venn 2.1, this is the first thing that pops up. There's lots of tools to make Venn diagrams. I found this to be the easiest one if you're just doing basic Venn diagrams. So now I go to list 1. I'm going to paste in my values, and I'm going to call this was leaf litter. Okay, and I'm going to jump back to the spreadsheet, which you can't see, and I'm just going to choose the data from the house plant. I'm going to copy and paste it like I did, and I'm going to paste it in here, and I'm going to call this house plant. Okay, and I'm going to jump back to my Excel file once more, and I'm going to take Peninsula soil, cut and paste, jump back, paste it in. Okay. And now I can see the Venn diagram is forming over there. There's a couple parameters I can change. I'm going to make it colorful. Okay. So now I can see the colors. I don't like serif, so I'm going to do sans serif, whatever. You can fiddle around with these things a bit. But what this Venn diagram is telling me is what phyla are shared between these three samples, which ones are unique to a given sample, and so on and so forth. Okay. And you know, the lists of the phyla that are in these samples are over here. It looks a little crazy because I have a lot of empty spaces, which is fine. It doesn't look so pretty, but it still maps fine. And what I think is really cool, and what I like to have the students do, is oftentimes you have a bunch of samples and you want to say, well, what's the core? What's shared in all of these? And so if you just click on the number, if you look over here to the results, it tells you that in leaf litter, houseplant, and peninsula soil, these are the phyla that are shared. Okay, if I want to look at which ones are shared between leaf litter and peninsula soil, I can see that it's just these three phyla. And you can also right click and you can copy this image and download it. So I think that that Venn diagram works nice. I prepared another one where I did it, I think I did this at the level of genus. It can get pretty complicated. There are some limits. The things that I don't like about it is it's limited to 4. And the circles are not, or the ellipses are not proportional. Okay, so it always looks the same, even though this is only 42% of the phyla are shared. That should be a lot bigger space than what's actually shown. So the circles aren't proportional. But in terms of doing something that's fast and easy, this works nicely. Any questions on the Venn diagram?
Would it make sense to average or combine multiple data sets from the same site?
Yeah, absolutely. So this would be more informative if I'd taken my leaf litter samples and merged and gotten an average, and if I'd done that the same with those. But the other thing that's nice about this is I could have shown my 3 peninsula soil samples, and I could have seen, what's the variation within samples that are from the same site, what's my core for those 3 samples. So it kind of depends on what you're doing, but yes, definitely averaging and presenting it as one value if you want to compare across sites makes a lot of sense. Okay, so any other questions on the Venn?
Does it make sense to subtract your empty column?
You mean the negative control, like my contaminants, or?
Yes.
Yeah, so if you know that you have a negative control, you want to remove those things, then yeah, you can do that. Again, the key here is getting it set up in Excel, and then you just paste in the names, and it's a piece of cake. And you can go brute force and you can just type everything in manually, but that's that's pretty hardcore. So that's why I like to do it in Excel first, and then I can just cut and paste. Anything else? Okay, so I hope that you really like the heat maps and Venn. I think it's cool. It's a lot of fun to do with the students, and it really gets them into the data. But I don't want to just blow ahead and kind of rip through the PCOA with QIIME 2 or Microbiome DB. I want to make sure that we have a chance to talk about assessment. And if we have a few minutes at the end, I'll jump back and show you these things. But we have video tutorials for doing the PCOA plots in QIIME 2 and the Microbiome DB. I think we have a pretty rough video, but I can make a better one. So, and again, if these are things that you want to follow up on, we'll have a poll at the end. We can have a subgroup that meets if you want to say, I really want to move ahead with the Microbiome DB. But if we have a few minutes at the end, I can try to show it to you, but I don't want to kind of miss out on some of the other elements that we wanted to cover. All right. So I'm going to switch over to Avram, or is that? I think that's the case.
Yeah, you can.
That's good. That's good. And then we can start at 17.
Are you going to share your screen, or do you want me to share? And
You go ahead and share your slides, Avram, and or Theo, and we'll tell you next slide, please.
Yeah.
Okay. If I find them.
There you go.
Can everyone see?
That's good. Right. If you can just go to the next slide. I just wanted to add a few minutes of the question, how do you measure learning in your students? How do you measure what the impact is on students' success? So if you want to go to the next slide, Theo. And this is more of a sort of social science way of viewing outcomes. So you're asking, so obviously your students have a cognitive learning in terms of Excel and data analysis and microbiomes, but what's the impact of that generally on student success? Because there's a great deal of interest in measuring the impact of research experiences on student success. So what is it possible to assess? And you know, if you're doing a very short course, you're probably not going to see much impact. But if you're doing a course that lasts a semester or longer, my guess is it's worth measuring the outcomes and seeing if the impact of the learning of the research experience itself sort of has some sort of effect, a ripple effect outside of the course. So, you know, these are the things that you can measure. For example, student grades. Will the experience in my class impact student academic success in the future in terms of grades? Well, the published work actually it's likely that you will not impact grades. However, it is likely, though, that you will impact STEM retention. Will the experience in my class influence my students' decision to stay in a STEM field? Or do they go on to do other research experiences? Have you stimulated their kind of research and innovation interest in the students? Will it motivate my students to do more research? Graduation, will it actually motivate my students to get on and graduate on time? And post-graduation impacts, will my students be more likely to do more research or attend graduate school or medical school? So go on to the next slide. So the question is, how do you measure these outcomes? And really there are two ways that are used. One is a longitudinal assessment where you would say, take your classes as the baseline and then start measuring the impact of your class on grades over time moving forward. And longitudinal assessments usually require control groups. So you might have to have a propensity score matched group, or students in other sections would also be possible. And a propensity score match group is a way for your institutional data folks to help you to find a student who has the same demographic background, the same kind of grades, the same gender, but has not taken the research experience. So that's longitudinal assessment, and that's best measured obviously over years. But there's also several qualitative assessment measures where you can measure student attitudes to science or college as a function of taking your class. And this can be quite important because it can really assess whether students feel more like they belong in college as a function of taking a research experience, which in fact is a common outcome. And there are different ways of using qualitative, there's different instruments for qualitative research, such as surveys with different kind of scales, numerical scales, or what they call Likert scales, focus groups, and interviews. Okay, next slide. So, okay, so you have some instruments, you have a plan, but it's also true that in a sense, if you're assessing the outcomes of your course, then you may need IRB approval for this. Institutional Review Board is there to protect the subjects of human subjects research. It's usually independent, a faculty-led committee. And you have to answer the following question to determine whether you'll need IRB approval. Is the activity that I'm performing a systematic investigation designed to develop or contribute to generalizable knowledge. Now, if you're asking the question, how does my class improve student success over the long term or student attitudes to science, you will need IRB approval. And in fact, the IRB will look at your consent form and your surveys and make sure that they conform to high ethical standards for human subjects research. The other question that you have to answer is, do I need a FERPA exemption? And FERPA is the way student records are protected. It's the Family Education Rights and Privacy Act, and it's designed to protect student records. But you actually almost certainly have an exemption to that as a legitimate education researcher because you are a professor in an institution of higher education, but you may need a letter from your legal department stating that you have a FERPA exemption for this to be added into your IRB application. So next slide. All right, and this is very, very quick. So I just wanted to tell you where'd you go from here, because I've really literally just touched on a few of the points. And there's three papers. There's one of my own papers, which was a mixed methods paper on longitudinal analysis and qualitative analysis combined. That was about student experiences in research in community colleges, where we looked at 400 students over several years. The second is Rodenbush et al., which is, I think, really one of the first very large studies from the University of Texas at Austin, where they looked at course-based research, which increases graduation rates and completion of STEM degrees. And the third is a review article by Lin et al. In Science on undergraduate research experiences, impacts, and opportunities, which, I think was a really good read because it really gets to the fact that there isn't actually a great deal of strong quantitative analysis of outcomes in terms of student success. And I've underlined two names in the first two papers. I underlined David Lopato in the first paper and Erin Dolan in the second paper. And they're two leaders in assessment of undergraduate research. And it's well worth looking up work by those two authors. We collaborated with David Lopato for surveys because he's an expert in constructing surveys to assess attitudes to science. And Erin Dolan is one of the leaders in this field of assessing impacts of research experiences. And yes, as Davida says, check out CureNet, which is a website. And I think if you start there, you'll be able to look at references and see sort of how to assess outcomes. And this has a great deal of value in terms of broader impact for NSF grants. Because if you're writing an NSF grant about your research and you want to integrate part of your research into a course for your broader impacts, you're going to have to do assessments. So figuring out how to assess the outcomes of the research experience in a classroom for your students is very, very valuable for broader impacts. And I'll leave it there. And anyone who has any questions about this can always email me directly. Thanks. And TR, I'll leave it there.
Okay, so in the last 5 minutes, we just want to briskly go through the next couple of slides. So we just want to remind you all that REMNet is a predominant, even though we've spoken about analysis, we really focus our efforts on curriculum design and integrating these kinds of things into your classes. So a lot of you are asking about curriculum design, a lot of you are asking about sharing of syllabi, and we're very open to offering a workshop, but I just want to show you, for many of you, you asked, how do you actually integrate this kind of thing into your classroom? We offer modular type approaches where you can kind of consider integrating any one of these parts or all of these parts into your course, right? So, and this is a standard micro course that Theo has been offering at his school. The analysis part for many of us has been optional up to this point, but the pandemic and everything else has shown us the utility of doing data analysis of this kind with your students. And Theo's presentation today has even shown us how you can leverage Excel. So they could be using an Excel workshop and looking at microbiome data as well. So there's opportunity for kind of interdisciplinary experiences as well. Next slide, please, Theo. And remember that for all of you, you don't have to focus on those big 4-year studies. You can also consider doing smaller studies yourself and publishing your course. And so we hope that in the future, if many of you are interested in this, you might be interested in doing a workshop on how to publish what you did. And you can aim for JMBE and smaller journals like JMBE and publish your own personal experience, what you did in your own course, and get that and put that on your resume as well. And it'll count towards promotion and tenure. Next slide, please, Theo. And remember, REMNet is a community, and this was one of the first exercises we made you all do. I'm hoping that you now realize that you're in a group of faculty who are mostly doing many of the same things. So as we said before, one of our goals is to grow the community with REMNet, and it would be fabulous if we could all come together and do a soil session and talk about what we're doing with soil across the country, and I think that'll be really great. Remember that you're not alone in all of this. Next slide, please, Theo. And you are now on our map. So we have mapped you, so we know where you all are. So you can expect to reach out from us, hear from us reaching out to you to find out where you are in the map. And when you click on this, you'll actually find yourself. So now you're on the map with us. So for next steps, next slide, please, Theo. We want you to consider joining us on CUBES. So CUBES is an organization that basically we're using for our community outreach and dissemination. So we'll be sending you all a link to ask you to join CUBES. CUBES has fabulous workshops on curriculum design and other aspects of biology education as well. But we have 5 working groups. So for many of you, you're interested, you may not necessarily have followed along with everything today, but you may want to know about wet bench, how to do the extractions. You might want to know more about the analysis. You might want to do access. You might want to do dissemination, whatever it is. There's an opportunity for you to get involved. And our final poll will be to figure out what kind of future workshops you would like to get from us at REMNet. So next slide, please. Our final poll. So if you can take this poll for us right now, and we may have time for it. I think Rachel is also going to put it in a form. So in essence, go to the forum, take the poll, and then we're going to develop our follow-up workshops on the basis of what you ask us to do. And remember that we're going to offer one-on-one sessions with you all for those specific nitty-gritty problems that you've had and come across during the course. Back to Theo.
Okay, thanks, guys. So our how's the poll feedback looking? I can't see the poll myself, but
we're going to do it as a form. So the form has been put in the chat.
Okay.
Tanya's saying that she wants to select more than one but can't. Okay, so we're going to figure that out. We might do a new poll that allows you to select more than one. So if you want to do more than one, we're happy to.
Rachel just revised it. So you can go ahead and submit it a second time and you can check all the boxes if you want.
That's great. Thanks, Rachel. Back to Theo.
Okay, so just a little bit of wrap up. As I said, we've started off the session with, in the first webinar with the FASTQ files and talking about the data and where the data comes from, uploading it into Nephele, and then within Nephele you have the 3 basic options of you can do quality control and do all the trimming and so forth, and you can do the analysis, and then there's an option for Explore. In that Explore, you can do pie crust. That's another exciting tool if you want to look at the metabolic potential of your microbiome sample. That's really cool. The Explore pipelines also give you the additional QIIME 2 outputs that we can come back to at a later point. And so then we started to actually get into the data with the outputs from the data 2 pipeline and looking at rarefaction curves and alpha diversity, the 2-dimensional peak-OA plots and bar charts and so forth we got into today. And then again, like we just saw, it's easy to take those data from the bar plots that come out in the QIIME 2 view and convert those into heat maps and Venn diagrams. What I didn't show is another kind of really easy entry-level thing that a lot of students enjoy is making a word cloud from your phyla, which is kind of fun. So these are kind of not too intimidating things that you can do with students if you're like we are working with an intro level microbiology course with students who are seeing this type of thing for the first time. So those are really visually engaging but not too technically difficult visualizations that you can do with your data. And again, we can come back for other sessions, or work with people individually to look more at Microbiome DB, which we didn't have time to get to, and some of the other resources. And hopefully we've been making this as clear as we can. Don't hesitate to reach out to us. Everyone's busy, and it might take a day or two for us to get back to you, but our goal at REMNet is to be able to work with people to solve their problems, even if they're just a issue that has something to do with your data set. So don't hesitate to reach out to us, and also take advantage of the REMNet community. Davida just showed us all the map. Look around, you might find that there's a core group of people in your area that can all work together. And so that, we certainly encourage that. And then finally, I'll just end up to thank you guys all for attending. Thank you to Rachel and Irene at ASM for getting this all set up for us. Thanks to Gina, who's done so much in the background that you guys haven't gotten to see. And yeah, we'll end it there. I don't have to rush off, but if there are some questions that people have, or if people want to kind of shout out the kind of things that they think would be valuable, even if you might have put it into the poll, I think we're open to taking a few minutes to do that, if that's okay, Rachel.
Got it. I'll finish up with some concluding thoughts. Put it in the chat, but pretty please, with cherries on top, give us feedback. We have a feedback survey for every webinar, and in order to keep programming like this through ASM, and we really need that feedback. And also the speakers really want to know as well, right? It helps them prepare their future events. And so please do the feedback survey. The second thing is we'd encourage you to, after you implement your microbiome analysis with your undergraduates, we'd love to hear about how it went at American Society for Microbiology Conference for Undergraduate Educators. I'll put the website in the chat. So it'll be online next year, the second week of July 11th through 15th. So do mark it in your calendars to come to the conference and share with us what you learned and what the students learned. Okay. So with that, I'll leave it to our speakers to entertain any other final questions or comments. Feel free to unmute yourselves, and I'll stop the recording